Hi, this is Teen Science, and today we're going to talk about prokaryotes. It means bacteria and archaea. They are everywhere around us. They live in seawater, rivers, lakes, in all kinds of soils, from desert to forest. They are in our homes, on everyday objects. They are also outside and inside the human body. The vast majority of them is harmless. A small number is useful, and a negligible percentage is unfortunately dangerous. This is the case of pathogenic bacteria. Prokaryotes were the first life form to appear on Earth billions of years ago. Let's talk about bacteria. They are divided into two main groups, gram-positive and gram-negative. The gram staining is a method invented more than 1 century ago and still in use. It requires 3 steps: primary stain with crystal violet, then ethanol decoloration, and counterstain with safranin. The result is a red-pink color from gram-negative and blue-purple from gram-positive. Easy. But why is this happen? To answer to this question, we need to investigate a little deeper into the structure of their envelopes. What separates the inside of the cell from the external environment is a series of layers. The first is the plasma membrane, the second is the cell wall, and sometimes there is also a third and a fourth level of protection. Let's start with the inner one, the plasma membrane, which is almost the same in both gram-positive and gram-negative bacteria. This is made of a double layer of phospholipids. But what exactly phospholipids are? Well, they're molecules that we can imagine as little magnets. They don't attract iron, of course, but water instead. And in particular, at one side, called hydrophilic head, they attract water, while on the opposite side, the hydrophobic tail, they repel it. Due to this peculiar characteristic, they spontaneously arrange in a double layer, with all the tails inward and all the heads outward. This is the basic structure of plasma membrane. It is associated with a wide range of proteins. Some of them lie on the inner surface, others on the opposite side, and they are both called extrinsic proteins. Many others instead pass through the double layer from side to side. They are called intrinsic proteins. These proteins have different functions, from sensing the environment to transport molecules through the membrane. They actually look like floating on a large sea of phospholipids, and for this reason it is usually referred as fluid mosaic model. The next layer is the cell wall. It is made of peptidoglycans and provides strength and rigidity to the wall's cellular structure. Here emerges the first difference between gram-negative and gram-positive, that explain why we get different colors. The peptidoglycans layer, in fact, is very thick in gram-positive and retains crystal violet so strongly that cannot be removed by ethanol washing. That's why they're typical violet color. The cell wall in gram-negative instead is extremely thin and cannot retain crystal violet, so when counterstained with safranin, it turns pinkish red. But what exactly these peptidoglycans are? We can imagine them as a bunch of pearl necklaces, where 2 types of beads alternate. The N-acetylglucosamine, that we can call for short NAG or just G, and N-acetyl acetylmuramic acid, also known as NAM, or just HAM. They are both sugar and form sequences of GM-GM-GM. All these chains are parallel to each other and are held together by lateral bridges between only the HAM beads.